Hello students, welcome back to Agriculture Form 4. We are still on mushroom production and now we are going to discuss on part 4, mushroom production. So far we have defined what mushrooms are. We have also looked on the importances of mushroom. We have also looked on the factors to consider when selecting the site for mushroom and factors to consider when selecting the mushroom species, as there are many species. And we have also discussed on the previous lesson on substrates, and we defined what substrates are and we've so far known that the mushroom, they are not just grown on a bare ground, but they are grown on a special material which are organic in nature, and these are the substrates. So we also explained on the previous lesson how these substrates are prepared. Remember, we said that these mushrooms they are very delicate. They can easily go bad if the place where we're going to grow them, they are not well prepared. So those substrates are prepared in 2 phases, as we explained last time. So since we have prepared our substrates, it means now we'll go into details on how best we can grow our mushroom. So on our today's lesson, you should be, by the end of this lesson, you should be able to describe how spawns are prepared, describe how mushrooms are planted, and describe the casing procedures. Be with me as we go through today's lesson. Substrate seeding or mushroom seeding. This is the process of germinating mushrooms from spores. Remember the life cycle of mushroom. It all starts with the spores. So remember, from the spores, those spores need to germinate. Once they have germinated, then they'll form what we call a hypha. So once the 2 hypha have joined, then they will come into what we call mycelium. They'll form mycelium. So from spores, then the other stage, life cycle stage, is mycelium. So the mushroom mycelium is propagated on the base of a steam sterilized cereal grain. So we just don't grow mycelium direct, but before we plant them, the mycelium, first of all, we propagate them at the base of a steam sterilized cereal grain, more especially millet. So once you mix the mycelium with cereal grain, or the mixture of cereal grain and mycelium, is what we call the spawn. So usually the farmers will grow the already made spawn. So spawn are mostly produced by specialized companies that supply growers with pure culture spawn. So what we grow, we do not grow direct the spores, but first of all, what happens that the spores uh, uh, will grow into mycelium. Then later on, once the mycelium have been formed, we mix with some cereal grains, small cereal grains, more especially millet. So once we have mixed the cereal grains with mycelium, we make this what we call the spawn. So the spawns are the ones which will broadcast in the mushroom house. So in some cases, apart from buying from specialized companies these spawns, already made spawns, but in some cases the farmers or the growers, they produce their own spawn. So it is important for us to go through how these spawns are produced. so that in case maybe we can be, as students, it's possible for us to gain skills and become a specialist on spawn production. So spawn preparation can be in 3 stages. Stage 1 is what we call culture medium preparation. On culture medium preparation, or culture medium is a nutrient jar on which mushroom fungi will be grown. So we try to find out an jar where uh, we try to grow our mushroom. So materials needed, we need potato dextrose, agar, substrates, and mud extra agar. So what's the process on culture medium preparation? First of all, you wash and weigh 200 grams of European potatoes. Then you boil them for 20 minutes in 1 liter water. Then later on, you drain the water. Then you add 1 liter of fresh water into the potato. So we have the jar here, and that's the jar which we can use. After that, after adding cold water, then you add 20 gram dextrose or glucose and 20 gram agar powder. Then you heat them until the agar dissolves. So you sterilize the agar in an autoclave or steam sterilizer. You let the agar cool on a laminar airflow table, and it is enclosed. This is an enclosed bench designed to prevent contamination of the biological sample. If that is done, it means we are okay. After culture preparation, then is it's stage number 2, which is tissue culture correction and preparation. So what are the materials required? So you need fresh mushroom, you need alcohol or spirit or ethanol, you need razor blade and forceps. You also need a cotton wool. You also need a bottle. So what are the procedures which you follow on tissue culture correction and preparation? First of all, we need to be very hygienic when you are doing a spawn preparation. That's why on stage number 1, we are mentioning of sterilizing everything which we are going to use, even to the point of boiling the potatoes, we are trying to eliminate the germs and microorganisms as much as possible, as these germs may destroy our mushroom. So when you are preparing tissue culture, when you are collecting the tissue culture as well as preparing them, we need first of all to wash hands. So first of all, you wash the hands. Later, you break the mushroom with clean hands to expose the gills. Then you remove the tissue between the gills. Remember, when we are discussing on the function of the gills, they are the ones which will produce the spores. So that's why we are getting the part of the gills. So you remove the tissue between the gills and the upper part of the mushroom using the forceps. Remember, the forceps which we are going to use as well should be sterilized. So you need to use the clean and germ-free forceps. Then you press the tissue in sterilized test tube and or petri dish using this forceps, and close the bottle near the frame, not to allow the germs to come inside. Then you close with a cotton wool. Then you press the petri dishes and the bottle in an incubator or a room temperature. After that, you go to stage number 3, that's spawning or spawn colonization. The materials which you need now, it's the sorghum, millet, as well as whole rice. Reaching this stage of spawning, it means you have already corrected the corrected the spores. So you soak all the materials for preparing the spawn in water for a day. Then you remove the water. Then later on you put the material in sterilized bottle. Then you sterilize the material. You let the content cool. Then you mix the tissue culture with the sterilized material. We call it inoculation. You inoculate. So it means we are done coming up with the spawn. So the planting of material of mushroom seed is called spawning, whereby you mix the millet with the mycelium. So this should be done when the moisture content of compost is 70%. Moisture content, then the temperature should be 25 degrees Celsius. Spawning is done by broadcasting 3/4 of the required spawn into the compost and mixing it thoroughly to the bottom. Then later on, the remaining quarter is broadcast on top of incorporated into the remaining top layer of the compost. So what you do is that as you are adding the as you are getting the compost or the substrates which are already prepared, you'll be adding little by little 3/4 of the spawn, the prepared spawn. You'll be adding to the substrate or to the compost. Then you'll be mixing thoroughly with the 3/4 of the substrate. So once you have done the mixture, mixing the spawn together with the substrates, then later on top of the substrate or of the compost, you finish the remaining quarter of the spawn on top. You just spread on top of the substrates. So you put them in an incubation house. So the doors and ventilators should be closed from spawning day to day 3. So ventilators are introduced on day 4, 30 minutes once in the morning and in the afternoon. Then colonization of mushroom fungi takes place within 12 to 14 days after spawning. So what do we mean if we say colonization of mushroom fungi? Remember the life cycle of mushroom. We mentioned of the stage of spawn, then the germination, the spawns will germinate, they'll form hypha, the hypha will emerge, then they'll form mycelium. From mycelium, the next stage was primordial mushroom. So once the mycelium have formed primordial mushroom, that's the colonization stage. So we're saying in order for primordial mushroom to be formed, it will take 12 to 14 days after spawning have been done. Then we do seeding procedure. So you pack the substrates into bags while adding planting spawn and tie the mouth of the bag. So this is done in an enclosed place where there is no air current. So you incubate the spawn bag in the dark place. Remember, when you are constructing the house for mushroom, we said that there are 2 houses of the mushroom which are used. We mentioned of the incubation house as well as the production shed. So in an incubation house is whereby the spawn bags are placed so that to make sure that colonization should take place. So incubate the spawn bags in the dark, then cover with a plastic sheet. So It will depend on the type of mushroom and how you have spread the spawn in order for colonization to fully take place. So it will depend also on the size of the bags. But the minimum is 14 days for full colonization to take place, depending on the size of the bags and spawning rate. So here are the bags. Remember, they are just hanged. They are just hanged. Those bags with substrates together with the spawns, they are hanged. So the colonized spawn bags are transferred to the fruiting house where they are opened after a day after light exposure. So make sure that you maintain the temperature at about 18 to 25 degrees Celsius, and the humidity should be 80 to 90% humidity, and this can be done by applying water several times, and air circulation as well is needed. So once colonization has taken place, mushroom colonization has taken place, then you transfer the colonized spawn bags from an incubation house to the production house or fruiting house. So in a fruiting house, these are the conditions. Make sure that the temperature should be between 18 to 25 degrees Celsius, and the humidity should be 80 to 90% humidity. We can achieve this humidity percentage by adding water, and also make sure that there is air circulation. Air circulation is very, very important as it will help to eliminate excess carbon dioxide. Too much carbon dioxide leads to failure to fruiting or development of deformed fruits. So here is the example of the picture showing the mushroom which is deformed. So one of the reasons why sometimes the mushroom fruits are deformed is because of too much carbon dioxide. So that's why air circulation is essential in as far as mushroom production is concerned, to make sure that there is mushroom development or fruit development, and also to make sure that the fruits, they are not deformed. Mushroom fruits are not deformed. Of course, we mentioned earlier that these mushrooms, they are grown on substrates. Substrates are organic, special organic materials where mushrooms are grown. But it is also important that we add a little amount of soil. We add a little amount of soil. And this little amount of soil which is added to substrates where mushroom will be grown are supposed to be pasteurized, are supposed to be sterilized. So the addition of moist pasteurized soil onto the substrate or compost is what we call casing. So students, we need to understand the term casing. We are saying casing is the addition of moist pasteurized soil onto the substrate or compost. So the soil which will be used for casing must be clay or loam subsoil, must be clay loam, and should come from subsoil. Why should the clay loam soil used for casing come from subsoil? Remember, in Form 2, we learned that on the uh, layers of the soil profile, layers of the soil. So we have topsoil, we have subsoil. And on the characteristics, we found out that the topsoil, there are several microorganisms. the microorganisms population is very high. But remember, when we are the place or the materials where we're going to grow our mushroom should not have a high uh, population of microorganisms. And subsoil is a true reflection of that. We find very little or less microorganisms under subsoil. So that's why we need to correct the subsoil. The soil should be coming from subsoil, but should be clay loam. And also low in organic matter, and the pH should be between 7 to 7.5 pH. So casing soil is obtained after removal of 10 to 13 centimeter of top soil layer. Most of the times, of course, depending on where you are going to get the soil, most of the times the top soil is not that deep. It's between 10 to 13 centimeters deep. So after removing 10 to 13 centimeters deep top layer of the soil, then later on it means you made subsoil. So after removing the topsoil, you get the subsoil. And casing soil is pasteurized by steaming at 70 to 80 degrees Celsius, at least for 15 minutes. Even though we have concluded here that subsoil has low microorganisms count, but the subsoil which you are going to use to plant, where you are going to plant mushroom, need to be pasteurized or need to undergo sterilization. So there is need of the soil also to be sterilized. So you can use steaming procedure. Make sure that the steaming should be at 70 to 80 degrees Celsius, at least for 15 minutes, while steaming to get rid of the remaining microorganisms. So this is done at the end of day 14 when fully colonized. So when they are fully colonized, when you are transferring the colonized uh, bags from the incubation house, and when you are transferring them into a production house, make sure that in that production house the substrates should be added with a little sterilized soil. So why casing? Casing soil promotes this and stimulates the formation of fruiting bodies and retain needed moisture for mushroom growth. So mushroom pinheads show off from day 16 after casing. So when casing is done, at least casing will promote and stimulate the formation of fruiting bodies. And apart from that, it will help to conserve moisture for mushroom growth. So two reasons for casing is to promote formation of fruiting bodies, and also to retain needed moisture for mushroom growth. So after 16 days, after casing, after 16 days, then you find out that the uh, pinheads will show off. So that makes the end of our today's lesson. Here we just emphasized much on how best we can produce uh, the spawns. Remember, the spawns are the ones which will broadcast for mushroom growing, and we discussed what the how spawns are made, and we have also discussed into details the phases or the stages how these spawns are produced. Though we have highlighted that these spawns most of the times they are produced by specialized companies, and farmers will just buy the already made spawns from those companies, then they'll grow in the mushroom house. But sometimes it is possible for the growers to produce their own spawns. So we have discussed on the procedures how these spawns are produced or are made. Then later on we have also discussed into details how we can plant these spawns and more especially in an incubation house. Then from incubation house, transferring, once they have colonized, we transfer them in a fruiting house or production house. But when we are transferring the colonized mushroom into the production house, we've also highlighted that casing should be done, whereby you add a little moist sterilized uh, clay loam soil onto the compost or onto the substrate. So in the production house, we add a little amount of soil, which is pasteurized, hence casing. If there are any questions, don't hesitate. Use the WhatsApp number displayed, or you can just visit Xulendo online so that you repeat the same lesson and fully understand the lesson. And these are the sources which we have on which this lesson have derived from. So you are free also to use these resources so that you add more knowledge on how mushroom is produced. So we are going to stop here until we meet on our next lesson, where we're going to finish off our topic on mushroom production. So till we meet again on our next lesson on mushroom production, goodbye.